Once again, welcoming you all to yet another session from BioPoint Stream the World of Bio. Here we are having the lecture 2 of Crash Course 2021 Molecular Basis of Inheritance. Topics that are going to be covered in this session are search for genetic material and DNA replication. So hope you all are perfectly ready for the session. Let's have the Josh. Give me thumbs up on the screen. Give me thumbs up on the screen if you are ready for this session today, right now. Okay, guys, so hope it's perfectly fine and let's move on. And once again, reminding you if anyone new is watching this session, do not forget to join the Telegram group where we have the daily session updates, regular discussion, daily polls, and where you can interact with me. So join now right at chi.me/neat_biopoint. The direct link for the Telegram group is given there in the description box. So guys, join right now with utmost pleasure. So once again, do hit the like button, do share it with your friends, and do not forget to subscribe the YouTube channel BioPoint Stream the World of Bio, and do not forget to press the bell icon so that you will be getting the notifications of the uploaded videos. Okay, guys. So hope you all are perfectly ready for the session today, right? So let's start today's session with the topic search for genetic material and experiments to prove which is the genetic material in uh what experiments to what prove which is the genetic material, right? That is what we have asked, whether it is DNA or protein. What is a gene chemically? The question that we have asked in the very first lecture, what is a gene chemically? Whether it is a DNA or it is a protein. So let's start today's session again asking the same question, what is a gene chemically? Whether it is a DNA or protein. So hope you all are ready, right? Okay. So the search for genetic material, the first experiment that we are gonna learn is the Griffith experiment. Griffith transformation or transforming principle experiment is the first experiment that we are gonna learn. This is an experiment conducted by Dr. Frederick Griffith. Dr. Frederick Griffith in 1928 using a pneumonia causing bacteria called as Streptococcus pneumoniae, using a pneumonia causing bacteria known as Streptococcus pneumoniae, right? So it was conducted by Dr. Frederick Griffith in 1928 using a pneumonia causing bacteria known as Streptococcus pneumoniae by taking another organism, mice. Okay, by taking mouse also along with that, he conducted the transforming principle experiment. Okay, Streptococcus pneumoniae, he used two stains to identify this what bacteria, pneumonia causing bacteria. That is, one is S type and one is R type. S type means smooth strain, that is virulent strain, that is smooth capsulated and virulent strain bacteria, that is disease causing bacteria. So virulent bacteria means disease causing bacteria, they cause pneumonia. So they has a polysaccharide mucus coat, that is smooth capsulated virulent bacteria. He has taken as S type, and as R type he has taken the rough strain bacteria or non-virulent bacteria with no mucus coat that do not cause pneumonia. Okay, and he conducted 4 experiments regarding this. He conducted 4 experiments taking this. So let's before moving on to this slide, let's say what were that 4 experiments, right? Let's say what were that 4 experiments. Okay, fine. So in the experiment 1, what happened, guys? In the experiment 1, what happened? In the experiment 1, he take a live S-type bacteria, and put it along with the mouse. Then what happened? What happened when he does like that? The bacteria, the mouse got affected by the virulent bacteria. The mouse got affected by the virulent bacteria. In the experiment 2, what happened is that he has taken a heated killed S-type bacteria. Heated killed S-type bacteria and gave it in connection with mouse. Then what happened there? That bacteria doesn't got affected. Okay. That doesn't got affected. Okay, fine. In the experiment 3, what happened is that he has taken a live R-type bacteria and kept it along with the mouse. And what happened is that the R-type bacteria is non-virulent, okay, and it is rough and non-capsulated. Therefore, and this R-type bacteria along with the mouse doesn't have any effect on the mouse. Okay, it is not affected. In experiment 4, what happened? He take a live R bacteria and heat killed S bacteria along with the mouse. He kept it along with the mouse, and what happened? This got affected. Okay, he concluded that the live R bacteria can change its character in the presence of dead S-type bacteria, and this is known as bacterial transformation. But this Griffith's transforming principle experiment was not accepted. Griffith's transforming experiment was not affected. He just said that it can transform in presence of dead S. But we do not have why we cannot say that it is transformed into a live bacteria because of the presence of dead S. Okay, because we do not have any verified proof there, right? So that is not at all accepted, and this is known as the Griffith transforming principle experiment. Okay, then once again let me draw the structure of, now I am going to draw the structure of S-type bacteria and R-type bacteria. Okay, so S-type bacteria So S-type bacteria, I'm just gonna draw right now. So one layer, and now we are going to draw another layer. Okay, this is capsulated, right? Capsulated, and now what we have to do is we have to draw the DNA in middle, right? We have to draw the DNA in middle, and here, here, what we can say, what we can say, this layer is known as, this layer is known as, this layer is known as capsule, and this layer is known as, this layer is known as, sorry, and that structure is known as, this structure is known as DNA. Okay, that layer is known as capsule and that structure is known as DNA. Is that clear? Is that clear? Is that clear, guys? Comment me right now. Comment me right now. The first layer is capsule and the inner, this thing is known as DNA. Now let's draw the non-capsulated R-type bacteria. Now let's draw the non-capsulated R-type bacteria. Non-capsulated R-type bacteria. So in this non-capsulated R-type bacteria, there is no inner layer. Only one layer is present. Only one layer is present. Only one layer is present, and that itself has the DNA. That itself has the DNA. So on labeling, we will mark this itself as DNA. No other further layeral division. Okay, fine. Okay, fine. Okay. So in the experiment one, as we have discussed, and he has taken an S-strain bacteria and injected into the mice, and the mice die. How it dies? Because it got affected with pneumonia and it died. Okay. And when he again what just conducted an experiment, how did that mice die, he found that the living strain cells are found in the heart of the mouse, and then he take an R strain bacteria, an R strain type bacteria, and injected into the mouse. And what happened? The mouse is living because it is not affected. R strain is non-virulent. It does not affect on mouse. Third experiment, he has taken an S strain or S type. He killed the bacteria, and that bacteria, when injected into the mice, no effect, and that mice also alive, kept alive. Okay, and that he killed this bacteria does not have any effect on mice. After that, he just combined this. He killed a strain bacteria and live R strain bacteria, and he just injected into the mice, and then the mice die. And then the mice die, and he found that S strain cells, living S strain cells in the bacteria, and he therefore concluded that some transforming principle is transferred from heat killed S strain bacteria to R strain bacteria, and that enabled R strain to synthesis smooth polysaccharide coat and become virulent. This is due to the transfer of genetic material from what live R heat killed S bacteria, his type bacteria to live R strained bacteria. Is that clear? Is that clear? Is that clear? Give me hearts on the screen if it's clear. Give me hearts on the screen. Okay, guys. Okay, fine. Now we are moving on to the second experiment. Second experiment conducted by Avery, McCarthy, and MacLeod. Avery, McCarthy, and MacLeod. Okay, fine. So this Oswald Avery and McCarthy and MacLeod worked to determine biochemical nature of transforming principle in Griffith experiment. They purified biochemical like DNA and RNA, the proteins, DNA, RNA, etc. to form and from heat killed cells using suitable enzymes. So now we are going to explain this transformation experiment principle conducted by Griffith, and this explanation was given by Ono. Okay, fine. And this transforming principle experiment, the verified proof was given by, or this was explained by Avery, McCarthy, and MacLeod. Okay, so now let's check that. Now let's first explain, as usual, let's first explain, and after that, let's give away the way for slides. Right? So, hope first explaining and after moving on to slides is useful. So kindly utilize that. According to this principle, or according to them, that these three scientists, the DNA particles of heat killed S came in contact with live R. So what happened? The DNA particles of live S bacteria, sorry, the DNA particles of, oh my god, if I clear this, everything will be going right. Wait, okay, fine. So the DNA particles of heat killed S bacteria heat killed S type bacteria came in contact with live R bacteria. Okay, so that, and so the character will change. Then they explain that DNA has the ability to change character. Then what they explain? They explain that DNA has the ability, very important point, DNA has the ability to change character. DNA has the ability to change character, and therefore they concluded that DNA is the gene. DNA is the gene. Very important. This was our question that we have started yesterday. Right? And now we have found an answer. What is a gene chemically? DNA is the gene. Is that clear? This is the biochemical characterization of transforming principle conducted by three scientists, Avery, McCarthy, and MacLeod. Is that clear? Okay, fine. So now let's move on with the slides. They discovered that digestion of protein and RNA using proteases and RNases did not affect any transformation. So transforming substances was not a protein or RNA. Digestion of DNA with DNAs inhibited transformation. It means that DNA caused the transformation of R cells to S cells, and therefore DNA was the transforming principle. This was how they concluded the biochemical transforming principle. Okay, is that clear? Till now? Exactly until now, we have just started this session, but we have explained a lot. We have a lot of theory today waiting for you. A lot of theory today is waiting for you. Okay, fine. Okay, fine. So now let's have this Hershey Chase experiment. Hershey Chase experiment. That is the blender experiment, also known as blender experiment. So this Hershey Chase experiment. So first, who will explain? As usual. As usual. Yes. First, I will explain, and after that, we will give the way to slides. Okay, guys. So now we have the green board. Okay. So let's look forward on the Hershey and Chase experiment. On the Hershey Chase experiment, what is the significance of Hershey Chase experiment is that the strongest evidence of DNA as genetic material was given by Hershey and Chase on their blender experiment. On their blender experiment. Okay, guys, the strongest evidence for DNA as genetic material. So this experiment was conducted by two scientists. One is A.D. Hershey, A.D. Hershey, A.D. Hershey, and the other one, the second scientist is Martha S. Chase. Martha S. Chase. So these are the two scientists, A.D. Hershey and Martha S. Chase. So they conducted experiment in bacteriophage. They conducted experiment in bacteriophage. Bacteriophage. So which bacteriophage? They conducted experiment in T2 bacteriophage. T2 bacteriophage. T2 bacteriophage. They conducted experiment in T2 bacteriophage. And this, what is this bacteriophage? They are viruses. Bacteriophage are viruses that affect bacteria. Bacteriophage are viruses that affect bacteria. Okay, is that clear till now? Is that clear till now? And multiplication of bacteriophage, that is they multiplies in bacteria like E. coli. They multiplies in bacteria like E. coli. That is the phage attaches to the above bacteria, that is E. coli, and makes a hole by lysosome. Just a hole is made in there. The bacteria have their bacteriophage have their legs like this, right? They have their, sorry, oh my god, this is the problem while using this squares and writing. Okay, fine. So this bacteriophages have their legs like this, right? Again, just simply wasting our time, right? Simply wasting our time. Okay, fine. So they have their legs like this, right? Again, like this. Then here, like this, and here, like this, and they make a hole in their leg by using an enzyme known as lysozyme. Lysozyme, like this. They act here like this. This is known as lysozyme. Okay, okay. And through the hole, it inserts the DNA, and using bacterial ribosome, phage DNA produces several copies of virus. So by making a hole here, it inserts the DNA to the bacteriophage, and this using this bacterial ribosome, the bacteriophage DNA suddenly starts producing several copies of the respective virus. Okay, finally the bacterium will break down releasing the virus, and this cycle will repeat, and that is how the multiplication of bacteriophage occurs. That is how the multiplication of bacteriophage occurs. So this is about multiplication of bacteriophage. So now what we have to learn is the Hershey and Chase experiment. So let's read the slides first, then after that again I will explain it. Okay, fine. Hershey and Chase grew some bacteriophage viruses on a medium containing radioactive phosphorus P32 and some others on a medium containing radioactive sulfur S35. P32 and S35. Viruses grown on P32 got radioactive DNA because only DNA contains phosphorus. Viruses growing in S35 got radioactive protein contains sulfur. Have you understood anything? Have you understood anything? No, right? No, right? So let me say that and make it clear for you. Let me say that and make it clear for you. Pakka clear for you. Okay, fine. Let me again bring the blackboard. Okay, fine. So this experiment was conducted by Hershey and Chase. They selected two T2 bacteriophages. They selected two T2 bacteriophages, two T2 phages, and one phage. What was the difference between two bacteriophages? One phage protein code was radioactively labeled using S35 isotope. One was labeled radioactively labeled using S35 isotope, and other was radioactively labeled using P32 P32 isotope. P32 isotope. Now, so these two phages were allowed to multiply in separate E. coli units. Separate E. coli units. Okay. And before the complete destruction of bacteria, their colonies were collected and in two blender machines. Colonies were collected in two blender machines and agitated. So they were collected in two blender machines. So blender machines and agitated. Blender machines and agitated. Blender machines and agitated. So the mixture were collected into two test tubes, and in the first test tube, radiations were emitted from viral coat. Radiations were emitted from the viral coat. That we will understood when you look the figure. Okay, radiations were emitted from the viral coat. That is, if this is the test tube, if this is the test tube, till here we have the mixture, and here we have the bacterial cells. Okay, here we have the bacterial cells. Here we have the bacterial cells. Let me draw a line here. Okay, fine. So here you can see the radiations are emitted. This red line is not, he's here to remark this radiation, and here if you have the bacterial cells. Okay, so radiations are emitted from the viral coat in the first test tube. In the second test tube, radiations were emitted from the settled down particles. Here from the viral coat, radiations were emitted. Here from the viral coat, radiations were emitted. In the second test tube, what happens is that, let's check. what happens is that here you have the bacterial cells. Okay, have the bacterial cells, and in the second test tube the radiations were emitted from the bacterial cells. Radiations were emitted from the bacterial cells. Okay, from here in the first test tube the bacterial cells is remaining silent. Okay, and the viral coat emits the radiations, whereas in the second test tube the bacterial cells emits radiation and the viral coat remains silent. Is that clear? Is that clear? So this means that the virus introduced its DNA into the bacteria for its multiplication. So DNA has the ability to pass from one generation to the next, and therefore they too concluded that DNA is the genetic material. Is that clear? Is that clear? So let's move on with the slides. These preparations were used separately to infect E. coli. After infection, E. coli cells were generally agitated in a blender. So look here, they are generally agitated in a blender. Let me use another color. So they are generally agitated in a blender. That is, they are generally mixed in a blender to remove the viral particles from the bacteria. Then the culture was centrifuged to separate lighter virus and heavier bacterial cells. Then what happened is that, oh my god, bacteria infected some virus having DNA were radioactive. That is, DNA has passed from one virus to another. Bacteria infected with viruses having radioactive proteins were not radioactive, and proteins does not enter the bacteria from the virus. So this is how Hershey and Chase proved that DNA is the genetic material by the blender experiment. So hope it's also clear for you. Look here. This was their experiment. Fine. So once again, reminding you, if anyone new is watching this session, do not forget to join the Telegram group where we contact where we have the daily session updates, contact regular discussions, daily polls, and can interact with me. Okay, so with full pleasure, join right now. t.me/need_biopoint. The direct link for the Telegram group is given there in the description box, and you will be having an amazing platform for mentor doubt clearance with our Biopoint Telegram group. Okay, guys, perfectly clear till now what we have taught. Okay. So now we are moving to study the properties of the genetic material. Properties of genetic material. So before moving on to that, what you have to do, as usual, what you will do in order to make me clear that you have understood. Yes, give me thumbs up on the screen if it's perfectly clear and paka for you. Okay, guys, now let's move with the properties of genetic material. So a genetic material must have the following properties: ability to generate its replica, chemical and structural stability, provide the mutations needed for evolution, ability to express as Mendelian characters. So these are the properties required for a genetic material. Is that clear? Okay, fine. So reasons for stability: less reactivity of DNA and high reactivity of RNA. Yesterday itself we have discussed why or which nucleic acid is more stable, and less reactive, and which nucleic acid is highly reactive and less stable. Right? DNA is double stranded. Presence of thymine makes it stable. and absence of 2-OH, that is the hydroxyl group in second 2- strand is in sugar. Okay, this makes DNA more stable and less reactive, and reasons for mutability in RNA, that is due to less high reactivity, single stranded, presence of uracil, and presence of OH group in the 2- strand of sugar. Okay guys, is that clear till now? Okay, fine. So the RNA is unstable, and all this we have discussed. DNA strands are complementary. All these things we have discussed. So have a silent reading right now. The slide. Have a silent reading. Okay, guys. So hope you have read that. So let's move on. Okay. So for the storage of genetic sorry, for the storage of genetic information, DNA is better due to its stability, but for the transmission of genetic information, RNA is better. So for the information storage, DNA is better, and for transmission of genetic information, RNA is better. RNA can directly code for the protein synthesis, hence can easily express the characters. DNA is dependent on RNA for protein synthesis. So the two processes that we are gonna learn in the upcoming two classes are transcription and translation. Okay guys. So here you have the transcription in the next class. The most awaited lecture. Wait for lecture number 3. Okay guys. So here in the lecture number 4 you have translation. Okay guys, and the lecture number 5 you have human genome project. Okay, so as you can see, all your slides are prepared for the entire chapter. Just in order, just we have to take it, and as per time we will complete that too. So now let's move on with the RNA world. RNA world. So RNA, as we have discussed right at the beginning of this chapter, was the first genetic material, and it acts as genetic material and catalyst. Okay, it acts as a messenger. RNA mainly functions as a messenger. Essential life process, metabolism, translation, splicing, etc. are evolved around the RNA. DNA is evolved from RNA for stability. Okay, guys. Okay. So central dogma of molecular biology. So what is our next topic, guys? Central dogma of molecular biology. So the central dogma of molecular biology, CDMB, was proposed by a scientist known as Francis Crick in is was proposed by a scientist known as what Francis Crick during his lifetime from 1916 to 2004. Okay, 1916 to 2004. Okay, fine. So let's check what is that. Oh. It states that the genetic information follows from DNA to RNA and from RNA to protein. From DNA, the genetic material moves on to RNA via the process known as transcription, and from RNA, it moves on to proteins via translation. So in some viruses, the flow of information is in reverse direction, that is from RNA to DNA, and this is known as reverse transcription, and this is known as reverse transcription. HIV virus is one virus that can undergo reverse transcription. Is that clear? Is that clear? So now the next thing that I want to remind you is that in the description box you can see the link for the updated playlist of Crash Course 2021, where you have all the chapters that are taken till now. Where you have all the chapters that are taken till now for the Sweet 2021 batch for the crash course. Okay, Okay, and this molecular base of inheritance will not be coming under this crash course, because it will be moving on to a separate lecture or a detailed lecture of molecular bases of inheritance. A separate playlist will be provided and will be updated in the description box soon after the end of the entire chapter. Okay, guys, so do visit this playlist and utilize maximum and revise your chapters thoroughly, and the links of this play what chapters taken is being provided as per time in the telegram group as the revision link of youtube lecture. So hope you all just do watch it and do share it with your friends. So let's move on with next topic, dna replication. Okay, guys. The another important topic, DNA replication. So, what do you understand by the term replication? Comment me right now. What do you understand by the term replication? So, the next topic, DNA replication. What is replication? It is the process of duplication, right? Replication is a process of duplication. So replication is the copying of, what is DNA replication then? It is the copying of DNA from parental DNA, or the process of duplication of DNA is known as replication. It occurs during the S phase of the cell cycle. S phase of the cell cycle, and it undergoes via enzymatic process. It undergoes via enzymatic process. On what all are the enzymes? What all are the enzymes used? What all are the enzymes used for this DNA replication? What all are the enzymes used? Exactly, the first enzyme used is helicase. Helicase. The second enzyme used is topoisomerase. 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 The third enzyme used is primase. Primase. The third enzyme used is primase. The fourth enzyme used is DNA polymerase. DNA polymerase. The fourth enzyme used is DNA polymerase. And the fifth enzyme used is ligase. The fifth enzyme used is ligase. Okay, guys? Okay, fine. So Watson and Crick proposed the semi-conservative model of replication, and it suggests that the parental DNA strand act as template for the synthesis of new complementary strands. And after replication, each DNA molecule would have one parental and one new strand. Then, Matthew Meselson and Franklin Stahl proved a semi-conservative model. Proved a semi-conservative model of DNA. So, who proposed that? It was proposed by Watson and Crick, and who proved that? It was proved by Meselson and Stahl. Okay, fine. So Meselson and Stahl experiment. Meselson and Stahl experiment. So the process of DNA is replication of DNA. You have to learn. If you are starting to learn, you have to learn it in very detail, right? Very detailed part that we will be covering. That we will be covering. Okay, fine. So let's learn this replication of DNA by using Meselson and Stahl experiment. First of all, I will read it so that after my explanation, after this reading, I will do my explanation, and after that, surely we can move directly to the next part. Right? So they prepared a two culture media of E. coli. So who they selected the E. coli for the experiment because they multiply in every 20 minutes. E. coli multiply in every 20 minutes, and normally E. coli contains 100% N14 DNA. N14 DNA, but two strands are containing N14 DNA and allowed to multiply this bacterium in N15 medium for many times. They allowed to multiply this bacterium in N15 medium. So what was that medium? It was 15 NH4Cl salt. So N15 was incorporated into both strands of the bacterial DNA and DNA become heavier. So after multiplication, this bacterium acquired this N15 isotope in two strands and 100% DNA, 100% N15 type of DNA is obtained. Right? So other preparation contains 14 N salt. I am explaining here in between after reading one sentence. Okay, so just cooperate along with that. So other preparation contains N14, 14 N salt. Sorry, 15 N and 15, 14 N or 40 N14 or N15. Okay, we can read it in any way. Incorporated in both strands of DNA and became lighter. These two types of DNA can be separated by using centrifugation in CSCl density gradient. So weight of this DNA is calculated by CSCl centrifugation. So weight of this DNA is calculated by CSCl. CSCl. Cesium chloride. Cesium chloride. Cesium chloride centrifugation method, and the 100% N14 DNA traveled less distance, and the second DNA, the 100% N15 DNA travels more distance. So N15 DNA has more mass than N14 DNA. And this Michelson and Stahl experiment is also known as CSCl centrifugation experiment. CSCl centrifugation experiment. So I have completed my explanation. So let's check what is there in the next slide. So they took E. coli cells from N14 medium, right? N15 medium and transferred to N14 medium, and after one generation, that is after 20 minutes, they isolated and centrifuged the DNA. Its density was intermediate between N14 and N15 DNA. This shows that newly formed DNA, one strand is old type, that is 15N type, and the other strand is new type, 14N type. This confirms the semi-conservative replication. After second generation, that is after 40 minutes, there was equal amounts of hybrid DNA and light DNA. Okay. Okay. So this proves that DNA replication is semi-conservative. Taylor and his colleagues performed similar experiment on Visia faba, that is faba beans, using radioactive thymidine to detect distribution of newly synthesized DNA in chromosomes. It proved that the DNA in chromosomes also replicate semi-conservatively. Semi-conservatively. Okay. Okay. Fine. The machinery and the enzymes. Machinery and the enzymes. Next topic, machinery and enzymes. Enzymes we have discussed now, and that we are going to learn now again. Okay. So DNA replication starts at the point called origin or ori. ORI, ori. A unit of replication with one origin is called a replicant. A unit of replication with one origin is called a replicant. Okay. So during replication, the two strands unwind and separate by breaking hydrogen bonds in presence of an enzyme called helicase. So this helicase breaks the hydrogen bond. So look here. Look here. So this is how the DNA strand is, right? This is how the DNA strand is. On the origin of replication, the helicase enzyme act. The helicase enzyme act, and this starts to unwind this hydrogen bond and breaks the hydrogen bond and unwind the two strands. So unwinding of this DNA molecule at some point forms a Y-shaped structure called replication 4. So if you look here, you can see a Y-shaped structure, right? A Y-shaped structure called replication 4. Okay, fine. Then the separated strands act as template for the synthesis of new strands. DNA replicates in 5-3- direction. So the direction of replication of DNA is 5-3- direction. Deoxyribonucleoside triphosphates, that is DATP and DGTP, DCTP and DTTP. Okay, so what are deoxyribonucleoside triphosphate, also means abbreviated in the form DATP, DGTP, DCTP and DTTP. Okay guys, so act as acts as substrate and provide energy for polymerization Okay guys, okay fine. Then firstly, a small RNA primer is synthesized in the presence of an enzyme primase. In the presence of an enzyme, DNA-dependent DNA polymerase, many nucleotides join with one another to primer strand and form polynucleotide chain. That is the new strand. This is how the replication occurs. Don't worry, I will explain it after reading all these things. Okay, so let's check how far is left. Three more slides to complete this lecture. So during replication, one strand is formed as continuous stretch in 5-3- strand direction, that is continuous synthesis, and this strand is known as leading strand. And the other strand is formed in small stretches, that is Okazaki fragments. That is known as Okazaki fragments in 5-3- direction, and this is known as discontinuous synthesis. Okay. So the Okazaki fragments. Oh my god, again. Reading and reading every time my throat is drying and the words are just interchanging. Okay. The Okazaki fragments are then joined together to form a new strand by an enzyme DNA ligase, and this new strand is called the lagging strand. If a wrong base is introduced in the new strand, the DNA polymerase can do proofreading. That E. coli completes replication within 18 minutes, that is 2000 base pairs per second. In eukaryotes, the replication of DNA starts at S phase of cell cycle and failure in cell division after DNA replication results in polyploidy. So that's all for today. Before saying thank you from my part, let me explain what we have just read about in this topic of replication. We have only read, right? We haven't learned. We have just read. Okay. So, process of replication, it originates in the region called origin, or ORI. Okay. So eukaryotes have multiple origin, whereas prokaryotes have single origin. And at the origin, the first enzyme act, that is helicase. The first enzyme that acts is helicase. Okay. And this helicase cuts the hydrogen bonds and unwinds the strand. So all of you know, as the strand is like this, right? So this is how the strand is. And when this helicase act on the strand, it start at the acting at the point of origin, like this. It starts to unwind, that is, it just separates this hydrogen bonds. Okay, it just separates this hydrogen bonds between base pairs. I'm just marking the bond or base pairs. Okay, fine. So then topoisomerase act, that is, it prevents recoiling of two strands. So after the action of helicase, the next enzyme that acts is topoisomerase. Topoisomerase. Okay, fine. So it acts and it prevents the recoiling of the strands. Formation of replication fork, that is start synthesizing new strand. So what is this replication fork? They are the Y-shaped point when the topoisomerase and helicase act on unwinds this DNA. So new strand is synthesized by DNA dependent DNA polymerase. So DNA dependent DNA polymerase. No need of writing, right? Okay, fine. But it needs a primer RNA for the synthesis of new strand, and enzyme primase provides primer RNA, and this catalyze DNA polymerase catalyze polymerization in 5-3- direction. 5-3- direction. That is the direction of replication. Okay. So and this new strand is always synthesized in this 5-3- direction. So one of the new strand is synthesis fast and continuously called the leading strand, and other strand is synthesis slow and discontinuously in opposite direction called lagging strand. Okay, so fragments of DNA in lagging strands are called Okazaki fragments, named after a scientist Okazaki. So after complete replication, DNA polymerase moves back and removes the primer RNA and adds DNA at this site of replication, right? So enzyme ligase joins this Okazaki fragments. After that, this enzyme ligase joins this Okazaki fragments together and forms the newly synthesized DNA as per the semi-conservative method of replication by Watson and Crick. Is that clear? So that's all for today. Thank you all. Thanks for your patient listening. It's me signing out from your favorite and my favorite BioPoint. Stream the world of bio. Bye bye.